Hello dear students, welcome to another video on sexual reproduction in flowering plants. We have already discussed about the parts of a typical angiospermic flower in our previous video. In this video, we are going to study the pre-fertilization structure and events. Pre-fertilization phase includes a number of structural and hormonal changes that lead to the differentiation and further development of floral primordia. These floral primordia give rise to floral buds which take the shape of flowers. Thereafter, the floral buds, sepals, stamens, carpel and petals start to develop at the tip of the receptacle or thalamus. Within a typical flower, male and female reproductive structures differentiate and develop. The male reproductive organ of a flower is stamen. It is also called microsporophyll. Each stamen has two parts, anther and filament. Anther is knob-like terminal fertile part, or we can say it is the pollen producing part of the stamen. A typical angiosperm anther is a bilobed structure connected together by a sort of midrib known as connective. Each lobe of an anther contains two pollen sacs or microsporangia in which pollen grains are produced. Now let us understand the bilobed structure of an anther with the help of a diagram. Here in this figure you can see top view cut section of an anther in which each lobe contains two pollen sacs or microsporangia. This is the first lobe and this one is the second lobe, having two microsporangia in each lobe. 1, 2, 3 and 4. Such anthers having two anther lobes and four pollen sacs or pollen chambers are called dithecous anthers or two-celled anthers. However, in some plants such as China rose, lady's finger, anther is having only single anther lobe and two microsporangia instead of four. Therefore, such anthers are called monothecous anthers or one-celled anthers. Let us understand the structure of microsporangium through TS of anther. In a transverse section, a typical microsporangium appears near circular in outline. It is surrounded by four wall layers: epidermis, endothecium, middle layers and tapetum. Center of the microsporangium has compactly arranged homogeneous cells called sporogenous tissue or microsporocytes. The given figure is TS of an anther. In this figure, you can see all four microsporangial wall layers and various types of tissues and their organization. This is the first lobe and this one is the second lobe. As each lobe has two microsporangia, therefore in total there are four microsporangia in each anther. This is called tetrasporangiate. They make the anther tetragonal or four-sided, as you can see in this diagram. The outer three wall layers, epidermis, endothecium and 2 to 3 middle layers, are protective in function and help in dehiscence of anther to release the pollen. The innermost layer tapetum provides nourishment to developing pollen grains. Each microsporocyte or sporogenous cell, which is present at the center of the microsporangium, undergoes mitotic divisions and produces a number of microspore mother cells or pollen mother cells. Each spore mother cell is diploid in nature, that is 2n. Each spore mother cell undergoes meiotic division and produces 4 haploid microspores. And this process of formation of microspores or pollen grains from a pollen mother cell through meiosis is called microsporogenesis. In this way, we have seen stamen, the male reproductive part. Stamen has anther and filament. Anther has pollen sacs called microsporangia, and microsporangium contains pollen grains called microspores. And these microspores are formed by the process of microsporogenesis. In our next video, we will study the process of microsporogenesis. Thank you.